Welcome. The webinar will begin in a few minutes. Welcome. Our webinar will begin in just a few minutes. Welcome. Our webinar will begin in just a few minutes. Welcome to the Harnessing Transformational Technologies webinar series, hosted by Los Alamos National Laboratory, the National Academy of Sciences Committee on Science, Technology, and Law, and Texas A&M University. Today, in the first of our webinar series, we have a renowned set of speakers who will offer their insights and expertise on the transformative potential of the COVID-19 pandemic. This is an unclassified event, so please refrain from making any classified comments or remarks. This event is being recorded along with the Q&A and chats. A link to the recording will be sent to all registered participants. We have attracted a large audience today. There are well over 700 participants registered for this webinar. To allow attendees the ability to focus on the speakers' presentations, we have disabled audience audio as well as the chat feature. Instead of using the chat feature during the presentations, we encourage you to send your questions through the Q&A feature of Zoom, which you'll find at the bottom of your screen. Our moderators will either answer your question privately or present those questions to the speakers to be answered during the webinar. We will try to answer all of your questions during the event. You may notice on the video that the presentation is sitting on top of, or the presenter is sitting on top of the presentation. To change this, you can minimize the video, or at the top of your screen, choose View Options, and then navigate down to the side-by-side mode. This will place the video next to the presentation. Now, I'd like to introduce our first speaker. Please join me in welcoming John Sorrell of Los Alamos National Laboratory. John is the Deputy Laboratory Director, or Deputy Director for Science, Technology, and Engineering. Welcome, John.
Thanks, Paula. As Paula said, my name is John Sorrell, and I'm the Deputy Laboratory Director for Science, Technology, and Engineering at Los Alamos. I'm standing in today for my boss, Lab Director Tom Mason, who both sends his regards and his regrets due to an unanticipated schedule conflict. Welcome to the first in our webinar series on harnessing transformational technologies. When we first conceived an event on harnessing transformational technologies, we imagined a normal several-day workshop in Santa Fe. But it would seem that the current pandemic and our associated pivot to a virtual world made a self-fulfilling prophecy of our thinking on potential transformations. Almost 2 years ago, researchers at Los Alamos partnered with the Committee on Science, Technology, and Law at the National Academies of Sciences, Engineering, and Medicine to plan a conference on transformational technologies. We're delighted that our partnership with the National Academies continues for today's event. We've pivoted our format to a series of shorter webinars, and today's event focuses on COVID-19 and opportunities to harness this pandemic. Our discussion will not only address the science and security aspects of the pandemic, but also explores potential ethical, legal, and social implications. In the near future, our partnership will host events focusing on artificial intelligence and synthetic biology. The silver lining of today's virtual format is that we've been able to register hundreds of participants from Los Alamos, our parent partners, the University of California and the Texas A&M University System, and other labs and many members of the broader community, far more than any in-person workshop could have accommodated. Los Alamos was established in 1943 and ushered in the atomic age. Years later, Los Alamos played a key role in establishing both the Human Genome Project and high-performance computing. These are three powerful examples of transformative technologies that change the world. These technologies demonstrate the power of large scientific collaborations and big science. COVID-19 has already had profound effects across the globe. Once again, we are seeing an urgent demand for scientific collaboration to understand and manage this pandemic and its consequences. In closing, I want to express my personal appreciation to the organizing committee, which has representatives from organizations, laboratories, and universities from all over the country, and especially our colleagues at Texas A&M who are virtually hosting the event. It's also my pleasure to welcome our esteemed speakers and moderators, David Tatel, David Baltimore, Betty Korber, Mike Imperiali, Tom Inglesby, Alta Charo, and Anne-Marie Mazza. And with that, I'm delighted to welcome Judge David Tatel, of the co-chair of the National Academy's Committee on Science, Technology, and Law, and a sitting member of the United States Court of Appeals for the District of Columbia Circuit. With no further ado, welcome Judge Tatel.
Thank you, John. It's a pleasure to participate in this conference. The only thing that could make it better would be to be there in person with you in Santa Fe. The Committee on Science, Technology, and Law, which I co-chair with Dr. Baltimore, was formed 20 years ago. It focuses, as its name suggests, on issues that fall at the intersection of law and science. The committee is especially interested in examining how regulatory agencies respond to rapidly changing technological and scientific developments. Our members are scientists, university presidents, law professors, and judges. We meet twice a year to consider a variety of issues, some of which become topics for National Academy consensus reports. In recent years, we've explored ethical, legal, and societal implications of a number of emerging technologies, such as synthetic biology, artificial intelligence, gene editing, and neural organoids and chimeras. We've looked at scientific evidence that is admitted into court, including forensic science and eyewitness identification. And we produce, along with the Federal Judicial Center, a manual on scientific evidence, which covers about a dozen fields of science and is provided to all federal judges throughout the country. The COVID-19 pandemic raises many new and challenging legal issues. Indeed, we've already seen demonstrations around the country against stay-at-home orders, business shutdowns, and mask requirements. And we are all worried about growing opposition to vaccines and its potential implications for a COVID-19 vaccine. But the fact is that over 100 years ago, in a case called Jacobson against Massachusetts, the Supreme Court addressed essentially these very same issues. In an opinion by Justice Harlan, the very same justice who famously dissented in Plessy versus Ferguson, the court rejected a constitutional challenge to the city of Cambridge's mandatory smallpox vaccination program. The court declared, quote, upon the principle of self-defense, of paramount necessity, a community has the right to protect itself against an epidemic of disease which threatens the safety of its members. With that broad principle in mind, the court concluded that Cambridge's compulsory vaccination program was constitutional. Several features of the court's reasoning are noteworthy for our purposes today. The court treated quarantines as an easy question, observing that the court had already, quote, distinctly recognized the authority of a state to enact quarantine laws. And with respect to the vaccination ordinance, which was at issue in the case, the court didn't require extensive scientific evidence, relying instead on the city's findings that smallpox, and this is a quote, was prevalent to some extent in the city of Cambridge and the disease was increasing. That was all the court needed. That was all the court needed to sustain the constitutionality of compulsory vaccination. That it was prevalent to some extent in the city and the disease was increasing. Finally, the court deferred to the legislature's assessment of how to proceed in the face of scientific uncertainty. Listen to this language from the court's opinion. The possibility that the belief may be wrong and that science may yet show it to be wrong is not conclusive, for the legislature has the right to pass laws which, according to the common belief of the people, are adapted to prevent the spread of contagious diseases. Though the Supreme Court's guidance is instructive with respect to some of today's most talked about legal issues, lockdowns, quarantines, mask mandates, it leaves open many really tough questions, particularly where pandemic responses intersect with modern technology and epidemiology. How do constitutional and statutory rights, privacy rights, limit mandatory contact tracing? How far does liability stretch for businesses and individuals who are responsible for super spreader events? How can we ensure that regulators effectively balance safety concerns with expeditious approval of new treatments and vaccines? How should courts resolve requests for compassionate relief release. How should courts resolve requests for compassionate relief or Eighth Amendment claims related to a public health emergency? What labor protections are necessary and lawful to ensure worker safety and the ability to speak out about employer health practices? How far do federal and state executive powers reach in these circumstances? And has Jacobson itself lost relevance in the 21st century, as a federal judge just suggested just yesterday in striking down portions of Pennsylvania's pandemic response? I could go on, but I think the point is clear. The pandemic raises serious legal issues for our country and much food for thought for the Committee on Science, Technology, and Law. Thank you.
Thank you, Judge Tatel. Thank you, John. We will now begin our first moderated session. Please use the Q&A feature in Zoom to submit any questions that you might have. This panel will be moderated by Mike Imperial of the University of Michigan. Welcome, Mike.
Thank you, Paula, and thank you, David and John, for those great introductory remarks. And so our first session this afternoon is going to be on the virus, on SARS-CoV-2. And we're going to have two talks. And what I'd like to do is to introduce both speakers up front. You can find their detailed biographies in the agenda, so I won't spend a lot of time going into biographical information. Each speaker will present for approximately 25 minutes. We'll have time for a short question and answer after each talk, and then at the end of the session, we'll open it up for a broader discussion. And so our two speakers today are first, Dr. David Baltimore from Caltech. David is probably one of the most influential virologists in history. He's made numerous important contributions to our understanding of various viruses, from poliovirus to retroviruses, including HIV. And in fact, it was his work on retroviruses where he showed that these viruses that have an RNA genome replicate via a DNA intermediate won him, along with Howard Temin, the Nobel Prize back in 1975. And so David is going to talk to us, give us an introduction to virology, and then talk about how viruses can jump from one species to another and what the consequences of that are. And then David's going to be followed by Dr. Betty Korber, who's from the Los Alamos National Lab. Betty has a long-standing interest in viral evolution and has done many studies on various viruses. And most recently now, she has turned her attention to SARS-CoV-2 and will talk to us about some of the ways in which that virus is evolving and what the potential implications are for the progression of the pandemic and development of vaccines and therapeutics. And so I would like to now turn it over to David for the first talk. David?
Thank you. I should say before I start that the talk I'm going to give is quite similar to one that I just gave as part of an MIT course. If there's anybody who was on that webinar, you can take a rest during this presentation. Next slide, please. So I love viruses. In 1960, I went to graduate school. I came to MIT and started studying viruses, and I've never lost that focus in my research, although I did spend a lot of time working on the immune system that fights viruses. I never imagined that at the end of my career in research, a virus would rule the world, creating a pandemic of a sort that hasn't been seen since the flu epidemic at the beginning of the last century. In 1975, I was awarded the Nobel Prize for work in virology, And at that time, I considered myself very lucky. And I wrote about my luck. Next slide. In a talk in Sweden, and the thing I said that is most relevant is that a virologist is among the luckiest of biologists because he can see into his chosen pet down to the details of all of its molecules. The virologist sees how an extreme parasite functions using just the most fundamental aspects of biological behavior. Next. Now, the world of viruses is quite separate from the rest of the biological world. So the living world consists of single-cell organisms and multicellular organisms, of plants, of animals, of cold-blooded and warm-blooded animals, many other divisions. But layered on top of that is a separate world of very tiny creatures, viruses, that seem to have evolved from bits and pieces of the living world. Next, please. Originally, viruses were just defined as something that passed through a filter that held back bacteria or larger cells. And when we did isolate viruses, we found that their genetic material could be RNA or DNA. And that was amazing because in the whole rest of the living world, the store of information is held only in DNA. In fact, the majority of the smallest viruses use RNA as their genetic material, and it's mostly large viruses that use DNA. There is no agreed upon definition of a virus. I use the definition which is written here, tiny objects consisting of a protected nucleic acid core that lack ribosomes or energy generation and can duplicate themselves only by penetrating living cells and diverting some or all of the cell's macromolecular synthesis towards making more virus. Now, does that make viruses alive? It's a continual question of debate, and I don't have an answer for it, but I consider them a very important part of the natural world. Next slide. Viruses are very small, but on a log scale, which is shown here, they're about halfway between water molecules and mammalian cells. And there are very small viruses like polioviruses. There are bigger viruses like flu and herpes viruses. And then there are some megaviruses that have many, many genes and have only really been worked on and recognized recently. Next slide. How many viruses are there in the world? It's probably an uncountable number because every organism has its own set of viruses. There is a classification of viruses, and within that, we recognize something like 1,500 species of viruses that are organized in 10 fundamentally different families of viruses. And they're all parasites growing in some small range of host cells. Often, they only grow in one species, like the viruses of humans, many of which grow only in humans. Next slide. The simplest of viruses have a few genes, and others have even hundreds of genes. But the genes are insufficient to allow the virus to grow on its own. It has to get inside a cell where it can commandeer the macromolecular synthesis capacity of the cell and use it to its own ends. The viral genes that do exist are genes for the duplication of the genome of the virus That's, of course, critically important to keeping the virus going, and genes for the protective coat. And there are some genes that are involved in inhibiting the host cell so that the virus can take it over. Next slide. Viruses can use both the nucleus and cytoplasm of cells, and we have run experiments to ask how many genes of cells are involved in making the virus, and the answer is hundreds of genes. So the viruses fit themselves into the metabolism of the cell in many, many different ways. One of the key things viruses do against multicellular organisms like us is to inhibit the immune system so that the major defense that we have against viruses is one that the virus is constantly fighting. Next slide. Viruses multiply very fast. They can increase 1,000-fold in just 6 hours. And they have to be passed from one host organism to the next very quickly. Otherwise, they'll die off just due to environmental degradation. And that means that if we can stop the transmission for a little while, we could eradicate viruses, many viruses entirely. And we've done that. We've done it with an animal virus called rinderpest. We've done it with smallpox virus, and we're trying hard to do it with poliovirus right now. But occasionally viruses that grow in one species will jump to another species. We say they have emerged from the species they're in into a new species. And this gives the virus a wider host range, and therefore may be beneficial to the virus. Next slide. Once the virus gets inside the cell, it sees this very wide range of cellular activities. I'm not going to look at what these all are, but the virus intercepts each one in its own way and utilizes it to make more virus. Next slide. The viruses that scare us most are these emerging viruses because we haven't had experience with them before. They exist in the wild. They're generally infecting some wild animal like a bat. Bats very often are a host to the natural host to a virus. But almost every year, one of them emerges from the wild to infect a given domesticated animal or human, and then it spreads widely in those species. And you may not even hear about these because they occur so often. Next slide. An emerging virus then can be the causative agent of some new infection. And many of these viruses are transmitted by small animals, ticks, mosquitoes, midges, arthropods. And they may be transmitted for a little while and then it's a dead end and we don't see them again. Or they may develop into a stable relationship with a new host and become another part of the world that we have to worry about. West Nile virus is a good example of the latter kind. It's now a virus that we all have to worry about, but some decades ago, we'd never heard of it. So let me focus on HIV and SARS-CoV-2, the two viruses that are causing pandemics that we're living with. And both of them happen to be viruses that have no vector, that are just passed from one person to the next person to the next person. But let me make a few general points before I get to that. Next, please. Viruses are ubiquitous. This number of virus particles in the world is astronomical, 10 to the 31st, more than the estimated number of cells of organisms, which are around 10 to the 30th. So that would be about a 10-fold increase. But these are rough numbers. And these viruses presented tremendous diversity of morphology, of strategy. They're actually much more diverse than the multicellular organisms that we're more familiar with. Viruses, therefore, have a great plasticity and may be very capable of adapting in the face of the environmental changes that we're all living with today. Next slide. In fact, the genomes of viruses are not pure because viruses mutate so frequently that they're actually a genetic swarm. It may help them adapt to a new host, but no virus is a pure single genetic unit. Next slide. Viruses were first discovered at the end of the 19th century. We have learned over the 20th century that viruses can cause cancers, that viruses can grow in laboratories, and that we can see viruses when the electron microscope was invented in the 1930s. In the 1950s, we found that viruses have as their hereditary material both RNA and DNA, and that was a big surprise because we were just learning that DNA was found in all higher cellular systems. Next, please. Viruses can be classified, and I spent a while working on those classifications as they derive from the central dogma. Next slide. Central dogma of biology is the DNA replicates itself, can be copied into RNA and translated into protein. And in 1970, Howard Temin and I reversed the first step to show that RNA can be copied into DNA, and that thus there's a transfer of information back and forth. But from the central dogma, we could then think about how viruses multiply. Next slide. And we realized that there are a range of different viral nucleic acid, viral constituents that hold the information. Can be single strands of RNA, can be double strands of DNA or RNA. But what they all do is they generate messenger RNA, which is what is in the center there. In fact, we can change this diagram around, next slide, to say that there are really 6, in fact 7 classes of viruses, each of which has a different genetic system. And that's in the face of the higher cells that have only a single genetic system that's like those of DNA viruses. And so viruses probably reflect the very earliest stages in evolution, whereas the rest of the biological kingdom reflects only the very last stages of biological evolution. Next slide. The gene, I'm going to skip this and the next. Go on one and one more. So the scourges of our present civilization coming from the virological world are HIV causing AIDS and SARS-CoV-2 causing the disease COVID-19. Focusing first on HIV, it's a slow virus. It causes diseases that take a long time to develop, as AIDS does. AIDS can spread out in an individual over 10 or more years before fatality. But without treatment, HIV was a completely lethal disease. Now we have drugs for it, and so we can live with it, even if we were infected. It grows only in one kind of blood cell, the CD4 blood cell. It's the only cell to which it can bind, and it's the only cell in which it can get into the interior of the cell. Next slide. The virus can, why don't we just get on to the following slide? The virus can get into cells by binding to the surface of the cell, and you can see that up here. The virus binds to the surface of the cell. It then puts its core into the cell, which activates the reverse transcription process using enzymes that are in the virus particle. And then the genome is transmitted into a double-strand DNA, which goes into the nucleus of the cell and integrates itself into one of the chromosomes of the cell. And now what was a virus has become a set of genes of the cell. It's copied, just like other genes of the cell, into RNA, and that RNA encodes the various proteins that come together at the surface of the cell to make new virus particles that are released. So it's a relatively complicated life cycle, going from RNA to DNA back to RNA, and putting together many different proteins to make the final particle. Next slide, please. As a disease-causing agent, the HIV lentivirus causes the AIDS, and we have to ask then, where did that virus come from? Because we never saw it before the early 1980s. And in fact, it was first seen in the United States in the 1980s. And then we realized that it was already spread through Africa. We now know that there was a progenitor of that virus, which was endemic in African monkeys, and that it was spread from African monkeys to the great apes of Africa. And people who captured the great apes and butchered them were probably the first people to get this infection from the apes. And then it spread silently, really over decades, through sub-Saharan Africa, and then to the United States, carried presumably by an airplane. And without treatment, it was almost uniformly lethal. It's been the cause now of 76 million infections throughout the world and 33 million deaths. We have no vaccine for it, and it's still largely out of control, except that we can treat it now with drugs, but we have no vaccine to prevent its spread. Next slide, please. It's actually a poorly infectious virus. It needs to get into the blood system, and there aren't many ways to get there. But it finds the CD4 positive cells in the blood. Ways that it does get in there are by a needle injection, or by a cut in the surface of the skin or elsewhere in the body. But the major way that it is transmitted is through sex. It's lethal because it depletes the CD4 cells, and those cells are needed for our immune function. Without them, there are many organisms which infect us and can kill us. So this emergent virus of monkeys has adapted itself to transmission by sex in humans, where it is lethal. Next slide. Its major lethality is in Africa, but but it spread from Africa throughout the world, not so much in Asia, but very extensively in the United States and even in South America. Next slide. Pat, next slide. So now I get to SARS, but I'm actually, I see, running out of time. Let me just review what we know about SARS. Next slide. Next. It's caused by a coronavirus, and we'll hear a lot more about that from Betty Korber, and you've almost inevitably heard about it from the press over the last few months. Next slide. It's a virus that grows in a very simple life cycle. It gets into cells by binding to a receptor. It multiplies itself maybe 1,000-fold. It exits from the cell and then spreads to other cells and does all of that in a few hours. Next slide. There are 4 coronaviruses that cause a mild disease in humans. They're just common cold viruses. There are 3 that cause serious disease. The first was the one that caused the original SARS disease that was stopped by rigorous case isolation. It's one of the great triumphs of public health. There were only 8,000 people infected by that. There's MERS virus, which is found in camels and can get into the deep parts of our respiratory system, is highly lethal and is still around. And then there is SARS-CoV-2, which actually causes a mild lethality, but it came into our world with none of us having any protection against it, and therefore can spread to all human beings and is in the process of doing that. Now, why are these viruses so different from one another when actually, if you look at them in molecular terms, they're pretty similar? That's a question which people are working on, which we don't have a clear answer to. Next slide. The important part about this virus is that the symptoms that people get are a function of age. So very young people, who generally are the ones that transmit viruses in our society, get a very mild infection and it doesn't seem to be transmitted largely from these young people. It's the older people who would be protected if the virus were in an equilibrium with our society, that are seriously affected by the virus and that transmit it to each other. So very different sort of transmission scenario from what we're used to with things like the other common cold viruses and a virus like polio. We're in the process of trying to produce a vaccine so it can protect older people. I am particularly looking forward to that vaccine, as are many of my friends. Next slide. Next slide. So the recent pandemics caused large numbers of cases of AIDS, large numbers of cases of COVID-2, 1 million deaths, even though it's relatively mildly lethal. And the wonderful case of the original SARS virus, which was caught early and was completely isolated, and in fact is now not anywhere on Earth that we know about, but actually is somewhere hiding away and very likely to reemerge one day. Next, the question that we live with is what other viruses there are that are going to emerge. They're almost certainly waiting for us. We need to be ready for them. Historically, we've never been ready when one hits. Maybe we can change that. Next slide. Let me thank you all for your attention.
Thank you, David. So we have time for one or two questions specific to David's talk, and then we'll move on to the second talk. Paula, are you going to take over for the questions?
Hi. I have one question here from the audience, and that is, please give us your thoughts on the reality of a vaccine before the end of the year, and what are the major hurdles since we understand the virus so well?
The major hurdles are making a vaccine that we know works. We can make vaccines that look like they should work, and we've done that very effectively, but they have to be actually proven in human beings, and that takes time. So we have trials going on now that involve 30,000 and new trials up to 50,000 people, and it takes quite a long time to be sure that the vaccines that look like they should work actually do work. My guess is it'll take to the end of the year before we have the kind of certainty that we need about which viruses are really good, which viruses maybe don't work so well, and that the viruses don't have surprising negative consequences, as we've just heard about with one test in the last week. So I can't see us having a vaccine that we can use for large numbers of people until early next year.
I have another question from the audience, and do you have any thoughts on how to respond to scientific misinformation? For example, the document recently released from Li Mengyan claims that SARS-CoV-2 was created in a lab. I do not find the analysis convincing and am torn between engagement, actively disputing the document, or ignoring the work, not wanting to call more attention to it.
I think it's a mistake to ignore misinformation, especially when we have good information. And we have to be honest about how good our information is. No information in science is ever perfect. There are always questions that are open. There are always things we don't understand. And we have to be honest about those. But when there's misinformation around that's misleading people, then we have to face that and say it's wrong. And it's just going to continue because there are people who love to imagine scenarios, and there are others who buy those scenarios and then pass them on, particularly through social media.
Thank you, David. I think we're going to move on to Betty's talk now, and we can try to address some of these other questions in the general discussion at the end of the session. So Betty, I'm going to turn it over to you.
Okay, thank you. So first slide when you're ready. All right. So I'm going to be, after that lovely and broad overview of viruses that David gave to us to help us think about what they are and how they move through animals and into us, I'm going to be talking to you about how the virus might be evolving in the pandemic and the tools we have to understand that evolution and how it might impact a vaccine and how we can stay ahead of it, hopefully. And we developed some tools at Los Alamos to try to understand these questions. And in particular, we noticed, and others have noticed, that there is a mutation in the spike protein. The spike protein is that little protein that sits off the surface of the coronaviruses. So you've seen it in all the images. It's the thing that stands out, and it's how coronaviruses got their name, because it makes it look like a crown. But this is the protein that enables the virus to enter our cells. And so it's particularly important because it's important for infectiousness of the virus, but it's also really important because that's the best target for antibodies. So a lot of our vaccines are targeting the spike protein. So anyway, we have noticed there's been a transition in the spike protein, and I'm going to tell you about that and how we came to that conclusion and how we're following this globally. So if I could have the slide, next slide. We published a paper this August in Cell that discusses this particular mutation. And so that's the author list and really the credits. The people in blue are all people from Los Alamos who worked on this project. But also this project was led by David Montefiore, who is a person who studies antibodies and vaccine responses. And his laboratory is going to be a leading laboratory in assessing whether the vaccine is working well against the virus. And Dr. Erica Ullman-Sapphire, who's in San Diego, who's going to be studying the different antibodies people are using as possible therapeutics. So you might take an antibody from somebody who was previously infected with the virus and they still have some antibodies persisting. You might be able to get some really good antibodies from those people that could then help other people. So that's Erica's focus. And both Erica and David helped on this project and helped understand the immunological implications of this mutation. So on the left, you can see the graphical abstract of the paper. And what we saw and what we documented was a global transition from the original form of the virus. And in this talk, the original form of the virus that came out of Wuhan and circulated throughout the world, we're going to represent with orange colors. So it's a D amino acid at position 614 in spike, and it switched over to a G amino acid at this position. And you can see the transition globally in frequency. So the epidemic spread and was pretty well established in a lot of places with this original form. This new form came up, and then it really began to dominate the global pandemic. And at this point in time, it's mostly what is sampled globally. So it is the current circulating form pretty much everywhere. And what we found was that this virus in a pseudotype assay, which is a laboratory assay, is more infectious, and it actually does have some immunological consequences, but there is a happy surprise. It was actually more sensitive to neutralizing antibodies that we're trying to raise with vaccines rather than less. So I'm going to tell you about those things in the talk. Okay, next slide. This picture is a picture that comes from GISAID, which is this amazing repository of sequences that's based in Germany. And I took this slide out this weekend. And at this point in time, GISAID harbors 88,477 full-length SARS-CoV-2 genomes. So these are 30,000 base genomes. And it's really a remarkable, I think, testament to humanity that they've been able to do this. So these sequences come from laboratories all over the world. Clinicians and sequencers, experimentalists get together, they sequence the local virus, they provide it to GISAID really swiftly. So a couple weeks after it's sampled, those sequences start to appear in GISAID. And these are made then available to scientists all over the world, like my group at Los Alamos, but really all over the world, to try to study how this virus is evolving. And it's actually evolving very slowly. So there's only a few mutations that accrue. And what you're seeing in this picture from GISAID is something called a phylogenetic tree. And so every one of those little tiny lines coming out of the circle represents a viral sequence. And sequences that are really close into the center of the circle have hardly any mutations. And sequences that have a lot of mutations have a long, long line coming out. And that long line represents maybe 9 or 10 or 20 mutations in this 30,000. Ones that have really short little lines maybe only have 1 or 2 mutations. So this is the global data set as of this weekend. And if you look at the center of that circle, you can see what are called clades, which are branches in these phylogenetic trees. So they would be like in a family tree, maybe your family members that are more related to you. These are viruses that have RNA genomes that are more related to one another. And so they're grouped together in the tree. And what you can see if you look in that center part are these major clades or groupings. And you start out with the gray virus, which was the original form from Wuhan. And then you shifted to a group called S and the V. And those were clades that had a few mutations that were characteristic of them. Those have pretty much all gone away in current sampling. So the green and the gray and that little lavender color, you almost never sample them in contemporary sequences. What you sample are these kind of pinkish orange and brown ones, and these all have the 614 mutation in spike. So this is their way of representing the shift in genome. And then we have a couple of other clades that came off of this basic G clade. They're called the GR and the GH clade. And so they're like the G clade with a couple of more mutations. So this is all being tracked in GISAID, which is remarkable global effort to understand how the virus is evolving. If I could have the next slide. So what we found was the single mutation in spike, going from D to G at position 614 in the spike protein. It's just one mutation in spike, and Erica Ullman-Sapphire made this really very beautiful structural picture, which shows you where it sits in the spike protein. It was associated with a greater infectiousness in pseudotype assay, with higher viral loads in patients in their upper respiratory tract, but it was not associated with increased hospitalization. So it doesn't seem to impact disease severity, but maybe impacts transmission. And so I'm going to walk you through the data for these things over the next few slides. But the newer things that we discovered after the Cell paper, I'm going to also walk you through this data, is Drew Weissman in a vaccine study, along with David Montefiore, who tested his vaccine results, found that this new form is actually more sensitive to neutralizing antibodies. And we found that we think we understand why, because it has a preference for an open conformation, and that makes it both more accessible to antibodies, and it makes it more accessible for binding to its receptor on a human cell, which is called the ACE2 receptor. So we think we understand the whole picture. So this is my talk in an overview, and now I'm going to try to explain the details of how we came to those conclusions. Next slide. So this is actually figure 1 from our Cell paper. And I'm going to walk you through each part of it and also give you an update of where it looks now, because the data is accumulating so fast that when we did this study in May and early June, we now have tens of thousands of more sequences, and the story is holding up as we look at these additional sequences. So this is really the epidemiology behind our belief this is a more transmissible virus. So if we look at the top part first, if we go to the next slide, we're going to look at just the maps of the world, and we've got a site at Los Alamos called cov.lanl.gov, where anybody can look this up with the daily updates from GISAID data. If you look prior to March 1st, if you guys remember, I'm saying orange is always representing the early form that was like the form that came from Wuhan. And then you look at a 10-day period in the middle of March, you can see that there were established epidemics all over the world that were beginning to shift to this newer form over this 3-week period. And if we go to the next slide, now I've got an updated map that shows you prior to March 1st, we have the Wuhan form getting established all over the world, but this new form beginning to show up strongly in Europe. The size of a circle represents how many samples there were from that particular region of the world at the time this map was, time frame this map was given. So the circle size represents the number of sequences, and the proportion of blue represents how many were the new form. So you can see by March 11th through 20th, there's already a lot more of the blue form showing up in many places in the world. And then by April 11th to 20th, it was dominantly the new form, and only little Singapore down in sort of the bottom right 3/4 still has a lot of the new form. There are a couple countries that are older form. There are a couple countries that still carry the older form, but still by July, the dominant epidemic form everywhere that was sampled is the new form of the virus. So the reason that we believed even as early April that there was a transition going on and that this was probably getting selected for was that in every single country where we looked, once the new form was introduced, if you gave it 2 or 3 weeks, it became the dominant form. So if that had only happened 1 or 2 or 5 times, we would have believed that could have just been random events. But the fact is it was happening virtually everywhere we looked. So by early April and the time we published a bioRxiv paper that was not peer reviewed, we had at least 24 countries that were changing towards the blue form and no cases that were changing towards the orange form. So it was always moving in one direction, and it was that repetitiveness that caught our eye and what we were looking for actually with the computational tools we developed. If I could have the next slide. Now we're back to figure 1 again, and I'm going to walk you through part B and how we could see this with GISAID. So one of the things we did was we tried to look at every place in GISAID where you could see a transition in frequency in a particular mutation. And so we go through all of GISAID data and we look at every single location, which are usually specified. We can do this at a continent, country, state, or city level. And then we ask, are there any changes in frequency in these 2 amino acids in this time period? And I'm just going to walk you through one case of it. It's going to be Washington, King County, and I've got it circled in red. And so this is what we pick out of GISAID at every single geographic level. We pick out every site where there's a change. The change could either be towards the new form or towards the old form, but we essentially almost always see it only towards the new form. So to see a transition, you needed both forms to be co-circulating. So that was one of our requirements. You needed enough data to be able to statistically see a change. And you needed enough time for that change to occur. So any place that had 2 weeks worth of data where both forms started out to be co-circulating, and we had at least 15 sequences prior to the time both forms were co-circulating and after 2 weeks, every time we meet those criteria, we pull out that local geographic location. So you can see what happens down in this bottom right-hand part of it. We had the new form was introduced right around the second week of March. We waited 2 weeks and then we saw that it was almost entirely the new form. So this pattern happens every place in the world. We don't see very many exceptions. We did find 2 exceptions in the paper, which I'll show you one of those. It was just if we gave it more time, we saw the blue form emerging. So if I could have the next slide. This is now an update, and so this is by country. So this update came from this weekend, and if you look at the top, this is the G clade that carries that G614 mutation, and we see almost every country, not quite every country in the world, you have an increase in the G form over time when we look at all of the GISAID data inclusively. So 37 out of 40 countries are switching towards the G clade form. But within the G clade, remember I told you that we had 2 other subclades emerging within G clade. So this is the GR clade and the GH clade that GISAID is tracking. And what we see with those 2 subclades is that those shift in frequency, they go up and down. And if things are just random, this is what we'd expect. So if you had founder effects, lots of travelers coming in, a big super spreader event, that could happen with either form if both forms are co-circulating. And that's indeed what we see with these 2 additional new clades that came off of that original G clade that we've been tracking. So if it carries 614, it always increases, but these little subclades can bounce up and down. And this is the kind of pattern we would expect to see with random events. So this is why we didn't think this was just a random event. It was the consistency of the recurrence of the pattern at every geographic level. Next slide. So finally, I'm going to walk you through a focus on North America. And another kind of thing we can do at our public database, if people want to look up a certain region of the world or any mutation, it doesn't have to be 614, you can look at transitions. So in figure 1, I just showed you North America as it was sampled by May, but this is now an updated sample that I'm showing you through September on the right. And you can see that in the beginning, when we just had a few hundred sequences, they were almost all the original Wuhan form for North America, but that transitioned, and so the frequency of the orange form declines relative to the new form until we no longer sample the original form. And if I could have the next slide. So we can now break down North America and start to look at it in more detail. And so prior to March 1st, we had some areas that had pretty well established Wuhan epidemics, and all of those areas by March 24th to 30th had shifted, so they were much more frequently the new form. A place like New York only really started with the new form. But I'm going to focus on 2 counties in Washington where we had the transition into a very well-established epidemic. And this kind of well-established epidemic, I think, was recurrent all over the world. But I'm going to give you the details on these because they were really beautifully characterized. So Washington State's just done a remarkable job with sequencing. They have thousands of sequences in GIS-8, and you can look at this county level. When I have the little pink lines in them, those were the stay-home orders that were required by the governor. So at the pink line, you have stay-home orders implemented, and then that soft pink area is the following 2 weeks after that, where you still might have had some transmissions from infections that were occurring prior to the stay-home orders. So if I could have the next slide. We're going to break this down. And so for these 2 counties, at the time stay-home orders were implemented, there were 614 cases in Snohomish County and 1,170 confirmed cases in King. And then there were sequences from 33 and 161 individuals, and almost all of them were the original form. A recent study has shown that there were probably about 11 cases for every confirmed case in Western Washington at this time. So there were probably about 20,000 COVID infections in these 2 counties up through the time stay-home orders were implemented. And greater than 95% of these 20,000 cases were the original Wuhan form, if you believe that the sampling was random from the sequences and the known cases. Despite that, there was the transition that occurred. The transition kept occurring in King County, even after this stay-home orders were implemented had been implemented for weeks. So this were really unlikely to be due to just some kind of a founder effect or travelers coming in, because there were so many people already transmitting from that region. So this is the kind of thing that I mean by it took over even in very well-established epidemics. This wasn't only happening in Washington, this was happening in Asia, this was happening in Australia, this was happening in Europe, South America. So these kinds of transitions were happening and well documented throughout the world. Next slide. So this is just a quick overview of this. This result was very controversial, and a lot of epidemiologists sort of didn't like looking at it this way. They wanted to use the phylogenies, which I thought were a difficult matter for COVID because there were so few base mutations for using the phylogenies. This is why I chose to look at this through a transition in frequency. But part of, again, why we believe this was really important is there's the WHO map as of March 15th for confirmed cases. So you can see where there were really pretty well-established epidemics. And the COVID map shows that there was a lot of the original form in these established epidemics really all over the world. And despite this, we've had to transition to the new form virtually everywhere in the world at this point. So now the new form is the relevant form, whether it was increasing transmission or random events that caused it, it is the relevant form that's circulating in the world today. So is the most relevant form for spike vaccines. Next slide. This is just another tool that we developed, and I'm just going to say really quickly, I really like this tool a lot. It was developed by Nick Hengartner, who's a colleague of mine in my group at Los Alamos, and it looks at transitions over time on a daily basis. And so you can see the fraction changing every day from the original form to the new form. And I mapped a couple of these, Sydney, Cambridge, but what happens is if you get a statistical increase over time, then it shows up in the plot to the left, and then I did these examples. So those little plots with the dots in them, the size of the dot represents how many sequences were available, say from Sydney on that day. And then the proportion that were the new form is on the y-axis. So you can see the new form is increasing, and you can see the sampling. And Sydney and Cambridge were beautiful examples of the transition in form, whereas Yakima actually went the other way. So it was one of our rare exceptions. And I highlighted in the paper for transparency, we were trying to track the exceptions and understand them as well. And if I have the next slide. What we had at the time we did this sampling, which was on May 29th, from Yakima is shown on the left there, but what we have now on 9/12 is a transition to the new form. So Yakima apparently just took a little time to start the transition over, and so that's why it was initially an exception. If I could have the next slide. Okay, so before we published this, we wanted to have some kind of evidence that there was actually a phenotypic consequence of this change, that it wasn't just the epidemiological evidence that I just presented. And we teamed up with a wonderful group from Sheffield, England, led by Fushan De Silva, who's leading this clinical and sequencing effort. And he had roughly 1,000 sequences from people that he knew their status because he had the sequences, and he knew whether they'd gotten hospitalized or not. And he knew how heavy their viral load was by kind of a surrogate measure called a cycle threshold for diagnostic PCR. So diagnostic PCR is the way they were testing if these people had COVID-2 or not. And if it was easy to detect, that meant there was higher viral genetic material in their upper respiratory tract. So lower value means there was higher levels of viral genetic material. And the new form had significantly higher levels of viral load in this upper respiratory tract by this measure from 1,000 patients from Sheffield, where we could actually link these PCR methods to the form of the virus they had. So the next slide. So we had published this in a bioarchives paper before it was peer reviewed, and several other groups also started looking at it and published in archive papers, again, not peer reviewed, but they confirmed our findings. And since then, another group, Volts, has also confirmed these findings. So lower detection limits implying higher viral loads were always found associated with the new form. So this seems to be a very solid result because it's now repeated in multiple clinical sites. Next slide. But we didn't find any association between the status, whether it was the old form or the new form, and hospitalization. So this is the data that shows this, and this was also confirmed. This lack of association was also found in these additional studies that looked at the viral load and the status as well. So it doesn't seem to be making people sicker overtly, which is a wonderful thing, but it does seem like it might be possibly more transmittable from the epi evidence, and the fact that there does seem to be more virus in the upper airway where people are moving this virus out in the transmission. Next slide. There is some slight evidence that extreme disease might be associated with this new form, though. We found a very weak statistical support of a higher incidence of people who actually had to go to the ICU unit versus just hospitalized. So the most severe cases were slightly enriched for the G614. That's not a compelling statistic. But there has been another study, Becerra, Flores, and Cardozo, that when they looked at the patients over the spring in different countries, they found a higher fatality rate associated with number of cases that were of the G614 status at a country level. So there is some possibility still open that very extreme disease might be associated with this, but not the overall rate of hospitalization. Next slide. The next thing we found is it took a couple of months to really get the pseudotype assays for these viruses going well, because it was a new virus and a new spike protein. They had to adapt their experimental methods to get this. But Erica Ullman-Sapphire's group on the left and David Montefiore's group on the right showed that the spike protein is more infectious in their pseudotype virus assays. And the way you can see that in these plots, they use different kinds of cells, and use kind of different frameworks for their pseudoviruses, but they pop the spike protein into a backbone of another virus so they can study it safely and very, very quantitatively. And these are the tests that are going to be used to assess vaccines as well. And you can see that the blue spots are always paired higher than the orange spots. So these are the new virus versus the old virus in these assays. And this is showing that they are more infectious. And then once we had the 614 step going, Zhang, Farzan, and Ko and others had also shown this in a beautiful bioarchive study. They were the first to show this, in fact, that there was a higher infectiousness in the laboratory, and others have since shown this in lung cells as well as these slightly less natural target cells. So this is a very compelling argument that these are more infectious. And the pseudotype virus neutralization measurements have been found to be very highly correlated with authentic SARS-CoV-2 measurements in a recent study that I've cited on the bottom right. So it's a good predictor of live viral behavior. Next slide. We also had in our sub-paper, Dr. Ullman Sapphire had some sera from people who are convalescent SARS-CoV-2 patients from San Diego. And we found that there was very little impact on the sera antibodies on the neutralization of these viruses, although we did see a slight tendency for the G614 to be more sensitive. And the blue lines are slightly shifted to the right. And that slight shift to the right is an indicator that it's slightly more sensitive. But this was great because it either wasn't impacting at all, or if anything, these new viruses were slightly more sensitive to the antibodies that were coming out of sera from patients that had been infected. So this was a happy result, and we were encouraged by this. And if you go on to the next slide, we still felt it was really important to understand if this mutation impacted the susceptibility to neutralization of the two viruses, the original kind or the new kind, when you had vaccine-induced antibodies. And so Drew Reisman had been working with the vaccine I'm going to tell you about in a second, and David Montefiore with assessing his vaccine responses. And the reason I've highlighted Priyamvada Acharya and Sophie Gogo is because they have some structure work I'm going to show you as well, explaining the effect that we saw. But what we were afraid of was that many of the vaccines that are currently moving forward and are just showing lovely antibody responses, many of these vaccines are based on the old spike without the mutation. So we were concerned that maybe the new spike would actually not be as sensitive to these vaccines. And in fact, we found the opposite. So I'm going to make that case. There is an impact, but the impact went the opposite direction and is good news instead of bad news. So I'm going to show you the support for that in the next couple of slides. Next slide, please. Okay, so Drew had been studying an mRNA strategy for a vaccine platform. It's a wonderful vaccine platform. It makes very potent antibody responses that seem to be quite durable, and it's also very reasonably priced for manufacture. So it's got a lot of virtues. He was using either full spikes, or he was using just the receptor binding domain of the spike. So just the part of spike that binds to the receptor that lets it into cells. And those were the vaccines he was trying. And for the neutralization assay, David puts the spike into a backbone of a lentivirus, and it carries it safely. It's easier to work with in the lab. And he uses a luciferase reporter assay, which is very highly quantitative and reproducible, to see how well this pseudovirus does at getting into these human cells via the ACE2 protein. And if the antibodies can block that entry, and that's what we're going to be looking at. So the next slide. So this first study I'm showing you, Drew had done 4 groups of 10 mice, vaccinated them, and what he was looking at was dose and delivery in this study. So he's giving higher doses of the vaccine and using different strategies for delivering it. But we looked at the response, the vaccine response in these mice to both forms of the virus, the old form and the new form. And the new form of the virus is the blue one again, and you see higher bars than the orange in every single mouse, all 40 mice. And it's about a 5-fold effect. So the new form is about 5-fold more sensitive than the old form, even though the old form was used for the vaccine. So that's mice. Next slide. He had 11 non-human primates, which were macaques that were vaccinated. And you see the blue bars are always higher. So again, the form is more sensitive in these primates, and he had 5 humans. And again, in the human immune responses to the vaccine, the new form is more sensitive. So this was a little bit baffling because we expected, if anything, it to be escape mutation, and instead it's not escape, it's more sensitive. How's that occurring? If we look at the next slide, I'm going to skip, oh, actually, I'm not going to skip this one. I'm going to tell you quickly. This was also true with vaccine sera. So our friend Thushan De Silva in Sheffield, England, provided David with 70 vaccine sera. And you can see the blue bars are higher again in almost every case. So if people who were infected and made antibody responses, again, the new form was more sensitive. And it didn't matter if they'd been infected with the old form and the new, their antibodies were always more potent against the new form. Next slide. And finally, this is about monoclonal antibodies. So these are some really beautiful antibodies people have been working with that have been purified out from infected people. And this time it's a reverse. So blue bar being lower means it's more sensitive because it's the amount of antibody that's required. And in fact, we found that the monoclonal antibodies were always more potent against the new form as well. And even up to 162-fold more potent. So it really had a strong impact on some of these antibodies, but not all of the antibodies. It was antibody dependent how much this mutation impacted the outcome. Next slide. So why is this happening? Well, I told you briefly, I gave you a preview of this, but what's happening is the spike protein oscillates between different forms, and it has its closed form, and all of its little arms are down. And when that happens, it's hiding the ACE2 receptor that it uses to get into human cells, but it's also hiding the part of the virus that the antibodies bind to. So how much time it spends in that down position impacts how accessible the antibody binding sites are and how accessible the receptor binding site is. So the old form, if you look at the left-hand picture, this is from Priyambada Acharya's laboratory. She found that it was only about 46% of the time in that open conformation. So the ACE2 receptor and the antibody binding sites were masked more than half the time in the status of how this protein is sitting. But in the new form, it was 1 up 82% of the time. So it was really opening up access to the antibody binding sites and the receptor that enables it to enter cells. We did a modeling study at Los Alamos that was led by Nana Karen, and Rachel Mansbach was the postdoc that really led this work. And what Rachel and Nana found was that in molecular dynamic modeling, where they did an all-atom simulation on the supercomputers at LANL, and they looked at how the molecules moved and transitioned, they found also, just like Priyambada had found in the structural study, that the 1-up form was really preferred. So now we have a molecular mechanism that would explain what we're seeing in the epidemiology and the immunology and the infectiousness of the new form of the virus. So it was sort of a lovely story that made sense at all levels, and all of the evidence is pointing to a coherent explanation for what's going on. Next slide. So this is just a really quick timeline. On April 3rd, I called my friend David and I said, you know, every place I look globally, the G16 form is repeatedly increasing in frequency. And this is unnerving, and I believe it might be under some positive selective effects. And this was happening even in really well-established epidemics like I showed you for Washington. So we initiated the clinical collaboration at that point, and David started pseudovirus work to get not only the original form, but the new form going into pseudovirus assays. We also started the molecular dynamics and structural studies that you just saw, and all of these things took some months to implement. So it was a good thing we got going in April because it took several months to really understand these effects. Next slide. So by the end of April, we had what I felt was strong enough case to submit to bioarchives. We had 24 cases of the shift in different local regions towards the G614 form. We only found one exception, Iceland. We saw this in well-established epidemics, and we knew from Fushon de Silva's work with 500 patients at that time that it was associated with a higher viral load in upper respiratory. So there's a phenotypic association, and others began to test it. And so the next slide, by our time of our Cell paper final revisions on June 25th, we had 1,000 patients confirming this. The pseudotype experiments were done with Erica and David, and we knew that it was more infectious in the laboratory. And we knew that the seroneutralization sensitivity was not compromised, which we were very happy to see, and this had already been confirmed by one additional preprint. So this, at the middle point here, bioRxiv submission, this was really controversial, but I think it's become less controversial as time has gone on, and we have this corroborating evidence and more or less a complete study. But it was good to get going in 2.5 months. It took 2.5 months to get this experimental work done. Next slide. So we are keeping an eye on other mutations. None of them have shown the kind of strong impact that the 614 showed even early on. But there are sites that are still rare in the world that we're tracking and that we're monitoring constantly. So I'm going to skip the next slide. Like S447N is an example of this. S447N sits in the receptor binding domain and is of interest from other experimental work that's been done by other groups. Next slide. So conclusions, extreme repetition of the pattern of the shift geographically is what caught our eye. If you toss a coin 83 times and it comes up heads 80 out of 83 times, you're going to begin to stop thinking that it's random. And this is the kind of scenario that we were in. We didn't feel it was compatible with random founder effects because repeat introductions could not overwhelm really well-established epidemics every time. That kind of thing, I think, was an argument for it being non-random. And its increased infectivity has now been repeated in at least 4 separate laboratories. It's in higher viral loads in the upper respiratory tract, has been repeated in 3 different laboratories, and we think we have a mechanism thanks to nana-nana-Karen and Priyamvada. So next slide, final slide. So these are just acknowledgements, and I want to once again acknowledge how remarkable I think GISAID effort is as a global effort, and how grateful I am that so many people are giving their data so rapidly. I've never seen anything like this. And it's really going to enable us to keep track of the evolution of this virus. So we will know early on if things should be tested to make sure there are no changes in vaccine sensitivity as we move forward.
Thank you, Betty. So we're running a little bit behind. So I think what I'd like to suggest is that we move to the open discussion now. Paula?
All right, I have a lot of questions here, and the first one is there a difference between the initial large number of cases that we saw in New York, New Jersey, Connecticut back in April, which had a high case fatality rate, as compared to the current spread in Texas, Florida, California, which doesn't have as high of a case fatality rate?
So I can't really address that question very specifically, but I can say that I think we've gotten better at my understanding is through the press, right? Not through scientific literature. So I think that all of you, or many of you, have been reading that we've gotten better at treatment. We've gotten better at keeping people alive and know better methods. So I think that's maybe part of the answer. It's also fatality rates. Case fatality rates are a little bit difficult in different places because both the denominator and the denominator is really difficult. How many cases you have, how the testing is being done, and I think it takes a lot of very careful epidemiology to try to compare studies in that case. So things like the study that I showed you that showed a greater fatality with this mutation. That was done based on number of cases, and every country calculated the number of cases differently. So there's kind of a softness to that data that you have to keep in mind. But David, do you want to address that, or do you have thoughts?
I think the important parameter may be the age of the patients. I'm not sure, but remember that fatality is a function of age. And so you have to correct for that.
It's true.
Another question from the audience is the classification scheme described allows very nice groupings of like-minded life cycle strategy viruses. Can you speak to framing the current classification taxonomy in terms of evolution? And I think that one was addressed to David.
Yeah, I assume so. It doesn't work well for evolution because of the transfer of genes horizontally and because of the complicated evolution of any particular kind of virus. It's not a good measure. And you'll find that there are viruses that have proteins in them that look like they came from a virus of a very different sort, and probably did.
This one I think is kind of geared to Betty. Since the new G virus is more infectious than the D virus, should we consider modifying social distance requirements?
We've been living with this form now, I think, since early May as the predominant form. But I actually initially felt it was really important to bring the hypothesis that it was more infectious so people could consider that. The virus that was the original form was the virus we had when we really went into quarantine globally. I mean, the stay-home orders were being initiated all over the world, and they were in response to that virus. And then this new one emerged. We don't really know about transmission rates per se. What we know is that I think the epidemiological evidence supports a higher transmission rate, but I don't think it's conclusive. I think it supports it. We know it in the lab bench, and we've got a mechanism. But I do think that it really suggests, yeah, we need to be very careful. I think the fact that we could have a transition to a new form of the virus so quickly as well, I mean, literally 1 month, suggests that we've got to keep an eye on new mutations and that we don't want to be giving the virus room to move as we're getting ready for our first vaccines. And as they're first implemented, because if resistance to a vaccine starts to emerge, you know, we could also have transitions. And so I think all of this speaks towards social distancing and trying to use epidemiological tools to control the virus from an evolutionary perspective as well as just our own health. Yeah.
So there's several questions regarding vaccinations and asking the panelists if you have any reservations associated with taking a vaccination that might come out within this calendar year. So would you be willing to comment on that?
Do you want to go first, David?
Yes, I am personally very concerned that any vaccine be well tested so that I'm not a guinea pig for either the quality of the vaccine or the off-target effects of the vaccine. And so it needs to have been studied in very large numbers of people before I'll be comfortable to take it. But that's what's happening. And I believe that by early next year, we will have vaccines that have been tested that well. And I very much hope that they will not show any untoward symptoms?
That seems very sensible to me. I think I certainly agree with that. I think that there's a couple of concerns people have about, say, an antibody that might enhance infection or a T-cell response that might be negative, and there's some evidence of this in the past. And the vaccinologists I know are really thinking hard about that. So you can study this with the clinical trials, and you can also study this in animal models. And if you give them time to study this well, I think we can have a lot of confidence going forward. And I certainly know that's everyone's intent, to really characterize these vaccines as well as they possibly can before they recommend their use. But yeah, things shouldn't be hastened to a point that we don't know efficacy. We don't really know efficacy. We've got to have that established, and we have to know safety, and that's what these large numbers will tell us.
Yeah, I would just add that with respect to antibodies enhancing infection, I think if I understand the data that's come out with the use of convalescent plasma to treat patients, there's been no evidence in those experiments of
Absolutely.
enhanced disease.
That I agree with that, and I think that my sense of the feeling in the field from participating in a few of these kinds of Zoom conferences is that it's very unlikely. What I like is that everyone's being really extremely unlikely, but everyone's being really rigorous, so we're not blindsided, and that makes me feel comfortable. That kind of rigor. But yeah, I think it's extremely unlikely, and I agree with that.
Yeah,
That evidence with this virus, this SARS-CoV-2.
And as you both indicated, that's the reason why we need tens of thousands of individuals in these trials to get enough data to really be confident.
Right. Can I ask Betty a question?
Sure.
Sure.
In all of this sequencing, do you see any evidence of genetic recombination among different kinds of coronaviruses?
Thank you for asking. Yeah, the first study I got involved with was a study of how SARS-CoV-2 looks in relationship to other bat viruses and pangolin viruses. And this virus that came into us is a recombinant it has a pangolin ACE2, the binding receptor. It has a pangolin-like binding receptor in a bat-like background. But we don't have really super close bat viruses to this one, so we don't know exactly how it emerged. But recombination is an extremely clear part of the history of SARS-CoV-2, SARS-CoV-1, coronaviruses in general. If you look just between bats, so recombination, where you have in one host 2 viruses got in and mixed their genetic material, is a very common part and a very important part of coronavirus evolution. That said, we've tried to understand if it's also an important part of the pandemic evolution. And we do see cases of it that we can validate, but there's so few mutations. Again, it becomes a statistical issue of being confident. So there are a few cases we can be clear are likely recombinants, but it's hard to tell overall how important that is as a contribution to the pandemic worldwide, because there's just too few mutations to use the computational tools we have to look. But it's very clearly an extremely important part of coronavirus evolution, and the viruses that moved into people, I think had pathways enabled through recombination.
Thanks.
There are plenty of additional questions. Do you want to take our break, or would you like me to ask a few more from the audience?
How are we doing on time?
We are 41 minutes past the hour, so we are about almost finished with our break.
Oh, well then I think we should probably take a short break. And just as I'm skimming through some of these questions, I think some of them might actually be more appropriate for the next panel. And so maybe we can get to some of them there too. So Paula, how long do we want to break?
We are going to resume the session at 2:45 Eastern Time.
That would only give a 4-minute break. I think that's,
Yeah, can we go a little bit long? Give people 10 minutes?
Okay,
Yeah.
we'll take 10 minutes and we'll resume at 2:55 Eastern Time. I appreciate everybody's thought-provoking comments on this from this panel.
Okay, great. Well, thank you again to David and Betty for these great talks. And we will resume again at 2:55, right, Paula?
Yes, at 2:55.
Okay, great. Thank you.
To our Harnessing Transformational Technologies webinar. Our second panel will be moderated by Anne-Marie Maza of the National Academy of Science Committee on Science, Technology, and Law. Please use the Q&A feature to submit any questions you might have during these presentations to the speaker.
Thank you, Paula. Good afternoon and welcome back to the second half of today's symposium. Today we have two wonderful speakers to guide us through a host of policy, ethical, and governance issues associated with efforts to respond to a global pandemic. Joining us will be Tom Inglesby of Johns Hopkins and Alta Charo from the University of Wisconsin. Each speaker will talk for about 25 minutes and then take a few questions from all of you, so I encourage you to use the Q&A function. As we approach the end of this session, we will then open the floor for questions from both speakers, concluding around 4. So let's begin. First, I am delighted to introduce Dr. Tom Inglesby, Director of the Center for Health Security at the Johns Hopkins Bloomberg School of Public Health. Tom is probably familiar to many of you, as during the past 6 months, he's appeared on the nightly news shows, helping the country understand the pandemic, mitigation strategies, and a path forward. Tom, over to you. Tom, you're still on mute.
Thank you. Thanks so much, Anne-Marie, for the introduction, and thanks to the hosts for this opportunity to speak to you all. So I'm going to speak a bit about some of the lessons that we could take from COVID-19, even though we're still living through it. And obviously we have 20 minutes, so this is a snapshot of the many kinds of things that we could learn, but I just wanted to share some of the things that I think are worth thinking about now. Next slide. So starting with what we were thinking back in January and February. This is an epi curve, one of the early epi curves published in the New England Journal. And there are many things to take from this, but a couple of points that are worth looking at. First, by January 3rd, the Chinese government had notified the World Health Organization and other governments about this new epidemic. And by January 8th, they had January 8th, the Chinese CDC had announced that this was a novel coronavirus. And then 2 days later, they shared the sequence. And a lot of other important things happened, but just so we remember, many things were already known at the very beginning of January. Next slide. By the middle of January, it was very clear to the whole world that the Chinese healthcare system was under incredible pressure, that their intensive care units were filling up, that doctors and nurses were coming from around China to take care of people in Wuhan. And this appeared to be something that had never happened before in China and probably never happened anywhere else in the world. It was pretty clear, mid-January to the third week of January, that this was an unprecedented event. And we see a picture here on the right side of a emergency hospital that was created by the Chinese government, which was called the Fire God Mountain Hospital, because they couldn't care for all the patients that they were seeing in their intensive care units. Next slide. And so over the course of January, what we saw in kind of a rapid series of announcements or observations was that human-human transmission of this coronavirus was occurring. We knew that by mid-January. Very high numbers of healthcare worker infections, although some of that wasn't entirely revealed in January, we began to see it. It became more clear with time. this virus, this epidemic was caused by a virus in a viral family that had caused wide endemic infections and major epidemics. We'll come back to that. David already spoke to this. Healthcare workers started to die in January, and that was cleared to the world. That wasn't able to be suppressed or suppressed information. And an unprecedented series of actions were taken by the Chinese government, some of which would not be reproduced by any other governments in the world, but they were a signal of what was going on. And we also learned in the third week of January that cases were appearing in countries around the world, mostly detected through airport screening. So we had a pretty good sense of what was happening in January. Obviously, we didn't know the full picture, but it was clear that this was an unusual, unprecedented, extraordinary event which had features of pandemics. Next slide. So the first lesson I think I would take from this in this discussion is that when we see early on that a disease has warning signs of a pandemic, we need to act accordingly. And what I mean by that was that by January, or certainly by early February, not just the US, but the rest of the world had sufficient cause to initiate a whole series of events, which eventually have happened, but in many cases happened slowly. We had the cause to initiate a crash vaccine and therapeutic development program, cause to transition to a large-scale diagnostic technology effort and implementation effort around the country, around other countries. We had cause to ramp up our healthcare system preparations, including preparations for PPE and for ventilators on scales that were unprecedented. We had a need for very clear, frequent scientific communication from government. And we had a need for a very clear strategy for travel and trade. All these things, I think, should be, if this happens in the future, and hopefully it won't, but we should presume it will, we should have all of these things ready to go and have a much lower threshold for action than we did as a global community. Next slide. So another kind of lesson. First of all, just kind of reflecting on what David said before about coronaviruses. We've known about coronaviruses for quite some time. They cause a series of endemic infections, responsible for as many as 25% of common colds. They're important and a very important viral family. We don't think we have long-lasting immunity from infection. Next slide. On top of the background of endemic coronaviruses, we've had two major epidemic coronaviruses that are well known. We don't need to go through them again here. But relatively recent history and well documented. There's no doubt that SARS-CoV-1 and next slide, MERS have had major epidemic potential. When you look back at SARS-CoV-1, many epidemiologists back at that time said that we got lucky to some extent that the disease did not spread more widely, that it was able to be contained when it was. It certainly had clear major epidemic potential and actually caused a major epidemic, but also potential pandemic potential. So in a rationally operating global health system, this would be enough for us to initiate, not only in the US, but in other parts of the world, a major vaccine development program that prepared for the possibility of pandemic. Next slide. We didn't do that. We did have some vaccine development projects, which I think, thank goodness we did. And largely they were funded by an international organization called CEPI, the Coalition for Epidemic Preparedness Innovations, which has helped us get a head start on a lot of the vaccine work that has been so valuable in the last 7 months. But I think the lesson that I think we should take from this now is that we should now establish a major pandemic vaccine development effort that relies on the creation of platforms, dedicated funding, dedicated staff, regulatory research that prepares the way for the rapid development of vaccine in the future. We're not going to know what that vaccine should be because we don't know what the next pandemic will be. But we could be further along in ironing out the process kinks and preparing the system the regulatory approaches, the trials that we need. And I think we're learning a lot about that now. We're doing, I think, an amazing job is happening now, but we need to learn what we can and see if we can make it even more efficient and see if we can move it up even every month that goes by without a vaccine in this kind of global crisis is the cause of thousands of lives lost, tens of thousands perhaps, and probably billions of dollars, if not $1 trillion a month. Next slide. So wanted to reflect for a moment on what our assumptions were about pandemics coming into this event. Starting with not an assumption, but something that was actually a model early on in March was the Imperial College model that helped convince the White House, the federal government, to impose social distancing measures or to call for them around the country. And you see on the left-hand chart, you see the model that became widely publicized, which anticipated as many as 2.2 million US deaths if no social distancing measures were undertaken. And you all can, this is a publicly available report if you want to see it, it's referenced at the bottom. This is Imperial College. If you don't know, if you don't study models for a living, they are one of the most well-respected modeling groups in the world. And they also modeled that a series of social distancing mitigation efforts could dramatically diminish the overall size of the curve and slow the curve down. So what have we been thinking about before our pandemic in terms of these issues? Next slide. There had been an effort at the US CDC to prepare for pandemics. And one of those efforts was a report in February of 2007, which talked about the idea of what they called back then community mitigation, which was, I think, largely a synonym for what we now would call social distancing. Another term people have used is non-pharmaceutical interventions. But what they showed was, or what they modeled was that if we put in place a series of community mitigation measures, social distancing measures, we could overall delay the peak, diminish the number of cases overall, and decompress hospitals, which became the guiding principles for much of the action, many of the decisions and actions that were taken back in March and April, and I think proved to be correct. Next slide. So I think that effort was very important. But if you look back here at that time, also in February of 2007, there was a lot of thinking about how to model the size of future pandemics. And I think this was a valuable exercise. It brought the community together, people who were studying pandemic influenza. Ultimately, they tried to create what is kind of analogous to a hurricane grading system with categories 1 through 5. In the end, when we tried to use that for 2009, it was very difficult to use because it takes a lot of time to figure out case fatality rate. It takes a lot of time to understand projected numbers of deaths. And so it wasn't operationally useful. And so we don't really use those categories now. But what's useful is to look at this table on the right, where you see what kinds of interventions were being considered back in this planning process in 2007. And you can see that when we got to category 4 or 5, they really weren't even thinking about that because it was such an extreme event. It was too far to the right. But if you look at category 4 and 5 on the left at the arrow, you can see that the case fatality rate there was 1% or 2%. In this pandemic, our case fatality rate, that is the number of people who get clinical illness and die from it in the US, is close to 3%. That doesn't mean it's the infected fatality rate, which is, if you include all the asymptomatics, those people who seroconvert, that's going to be closer to 0.5 or lower. And obviously, the case fatality rate varies dramatically based on age. It's very high for older people, very low for younger people, and somewhat in the mix in the middle. But the point here is that we didn't really even consider, in some ways, some of what were, some of the possibilities here. Next slide. And here, another way, in 2017, we had another pandemic planning effort, also, very useful. But if you look at down at the basic reproductive number, which is about halfway down the chart, and you see the different scales, we're no longer talking about categories, but just kind of a scale of severity. You can see that we were, in this planning process, we were planning for a reproductive number of pandemic along the lines of 1.8. Well, now we know that unmitigated, this pandemic, the WHO says, and CDC say that this pandemic, this virus has an unmitigated reproductive number that's more like 2 to 3. So again, this is really, kind of, I think this is just to tell, even with all of this, the best plan, probably the best pandemic planning efforts in the world at the time, the most comprehensive, it still exceeded our expectations. Next slide. And so third lesson here is just to say the past does not dictate the future. I think a lot of our pandemic planning efforts around the world have really planned around 1918. That's logical. That was our last major pandemic event of this size. But many of the assumptions that we held were wrong. I think there was a wide assumption that past pandemics might have presented the boundaries for future ones. And another, that influenza was really the serious pandemic threat, the main one that really ultimately garnered 99% of our pandemic planning energies. And that emerging infections, in fact, were not really, a serious concern for much of the world, but they were to be problems for other parts of the world. Related to this, we know that the world is changing, that the climate's changing, people are moving very quickly in the world. We have megacities, antimicrobial resistance. So there are lots of reasons to assume that what we see in the future will not necessarily reflect the past. And that's not just in the realm of infectious disease, but we often make that, there's a widespread mistake that the past predicts the future in many realms of human endeavor. And I think we should not assume that for biology. Our technology is changing, mostly as a power of incredible good for the world, but it's also so powerful that now it could be misused. It's widely distributed in the world. We now have the biology to actually create highly lethal transmissible organisms, or to engineer organisms to become more lethal, more transmissible. And there is now the possibility of accidents occurring in laboratories that could lead to trouble. So we have to incorporate all of that in our planning process around epidemics and pandemics for the future. And we need to plan and have very strong and effective global responses. We can't do this alone as single countries. It won't work. So we need a global plan. Next slide. So now thinking a little bit more about the particulars of what we've done since in response to this pandemic. This is just a very well-known and respected study that was published around the time of the pandemic planning effort in 2007. Howard Markle and colleagues studied the impact of what they called in this paper non-pharmaceutical interventions in the 1918-1919 pandemic flu response. Again, these are the same kinds of interventions that we talk about now when we talk about social distancing or physical distancing, school closures, banning of public gatherings, isolation, quarantine. And they found that in cities that implemented those kinds of interventions did better and recovered more quickly. So we have this kind of historical basis. Next slide. But coming into this pandemic, we didn't have any real lived experience with it. So I think many public health professionals around the world would talk about these measures, but I think there was some skepticism about whether they would work in practice and whether people would be willing to do them and whether leaders would be able to get people to go there. And so, now fast forward to January of 2020, when we saw what was happening in Wuhan, and we saw that there was an extraordinary rise in the number of critically ill patients in Wuhan, with peak day being about 2,300 people on ventilators in Wuhan at one time for one disease. If you scale that to what the US ventilator capacity, that's basically, if you normalize that per population, that would mean in a given US city, on average, that number of patients would have consumed every critical care bed in that city. So Wuhan, the Chinese government responded on January 23rd with an unprecedented lockdown, some of which could be, and would be emulated in other parts of the world, some of which would not be for specific reasons. China would take those and other governments would not pursue that approach. But what you see is that it took about 3.5 to 4 weeks to really turn that tide in Wuhan, but it did turn. And it began to consistently come down and came down steadily since that time. If you look at a sister city in China, Guangzhou, which is about the same size, on the same calendar, you see that Guangzhou experienced very few cases, critical cases or deaths. And what Guangzhou did differently was initiate its physical distancing and lockdown measures earlier, before they had a major problem in their city. So it kind of was two cities as kind of a little case control between different populations. Next slide. And then with the tincture of time, we could see that the measures that were being put into place in Wuhan, you could see that the effective reproductive number, which is the kind of the moving reproductive number, which accounts for geography and for interventions, changed and went down over time as interventions were put into place. And it's still a matter of debate and argument about which interventions made the most difference, but you could see as a group of interventions that the effective reproductive number came down pretty substantially, pretty quickly, and has stayed below 1 since that time. We do believe it's possible it's gone up above 1 in certain places for periods of time, but generally speaking, it's now, the number of new cases in China is quite low on a day-to-day basis. Next slide. You can also see the same phenomenon in Italy, which was the second part of the world, which had the obvious, one of the countries that led after China in terms of numbers of cases. And you can see that on March 9th, when a nationwide stay-at-home order was made, it still took a few weeks before the curve changed over time, and it has come down steadily since then. Next slide, please. So fast forward now to the US, when we were at the beginning of seeing cases in the US, and we see that in this particular week, this is the normal activity chart for US based on how people drove, it's a measurement of devices and the distances people drive. So fast forward, next slide, please. You can see that as you move towards green and blue, this is a measure of decreased movement of Americans. So by March 16th to 20th, in many parts of the country, we're now heading towards green, so traveling less than they had the week before, or even blue, which is 75% less than the week before. Next slide. So this is when, now you see what happened by the March 23rd, 27th week. And this is now governors around the country beginning to impose various social distancing measures, restrictions, stay-at-home orders. Next slide. And you see here a model that was done by Jeff Shaman and colleagues at Columbia. It was a preprint. I think it's still preprint unless it's been published. And their model concluded that if we had put in place social distancing measures 1 week earlier at the same scale, that as of May 3rd, there would have been a substantial number of reductions in deaths for the country. Again, that was a preprint, so we'll have to see how that comes out, but it's provocative in the sense that it really starts to measure the impact and speed of physical distancing measures. Next slide. And then again, this is just the US curve, which shows the impact of social distancing measures in April, the subsequent period of time after all states reopened, and then a gradually coming down and reimposition of some mix of measures around the country starting in August. I think many things are happening at the same time. So again, it's hard to really identify specific causality here, but you can see the trends from around the country and they do correlate, and they correlate with the experiences of other countries as well. Next slide. So the fourth lesson I'd call out here is just that in future pandemic planning, we should, even though we didn't really know what we were doing coming into this pandemic around physical distancing and public mask use, they should be the expectation and standard for the future. We do know that there are a lot of downsides with physical distancing, very high societal and economic costs. So we need to think through and study how this worked and what's happened. Physical distancing does not mean complete lockdown or people can't leave their homes safely. I think we know a lot now about outdoor risk being far less than indoors. We know that large indoor gatherings are particularly risky activity, and we should be doing everything we can to think about smart physical distancing for the future and to be thinking about public mask use in a way that we weren't preparing for before this pandemic. Next slide. I think just a few final short lessons, which I just would put in this mix, which is the terrible impact of COVID on nursing homes and prisons. Just to call out a few kinds of congregate living settings where things are very high risk. This is likely to be a serious matter for the future as well. So I think all pandemic planning efforts for the future should be, in any disease that's spread by respiratory transmission, we should be anticipating where the most difficult institutional spread is going to occur and plan accordingly. Next slide. I think what that means in practice is that we have to protect congregate settings intensely, and that is through PPE, that's through diagnostic testing strategies and approaches. It's through metrics that really study what's happening there, and planning efforts at the community level. Next slide. I think the other thing another brief lesson to finish on is just the importance of local and state leadership. Of course, this is a national crisis that the federal government's very important in what it does or doesn't do. But next slide. The story of the pandemic, of course, is told in 50 ways across the states, and then within their own communities. There's so much variation happening, even within a single state. This chart is from a Hopkins coronavirus testing site, which shows that even though the country, on the previous slide, the country was trending towards 5% test positivity, 32 states on the same day were really above that 5% positivity threshold. So you really need to get down into the details, and state leaders and their decisions will have an enormous impact on the pandemic. Next slide. And this again shows, this is now about 10 days old, but shows the huge range of conditions even now in the pandemic, with the Midwest being most seriously affected, but at various points along the way, the Northeast, California, the South have all been most seriously affected. And it shows, I think, many things, but certainly highlights the impact of individual decisions by leaders and by what the public is doing in various places to try to slow the spread of the disease down. Next slide. So I think we've covered this, state and local actions. And next slide, I think, which is maybe the last. Science. The role of science in this epidemic has been extraordinary. I think if this pandemic had happened 30 years ago or 20 years ago, it would be an entirely different, much more grim situation than we're dealing with right now. We have been able to use the science of epidemiology, genetics, infection control, the drivers of severe infection, diagnostics, and goes on and on. This is clearly a very partial list, but we're learning an enormous amount at speed. And I think we heard about that in the last presentation. One thing I think has not gone well that we should prepare for the future is the study of origin of this pandemic. It's been complicated by geopolitics and by a variety of things, but we need a better plan for getting to the origin of these events. But putting that aside, I think we have to take everything we're learning from science and be able to put it into action and make sure it's fully supported by our governments in the world to prepare ourselves for getting through this pandemic and for future pandemics. And I think I'll stop there. And Anne-Marie?
Great, thank you so much, Tom. I think there's a number of questions in the Q&A, and Paula, I'll hand it over to you.
right. There's several questions about applying current emerging technology into vaccine development. And I was curious that the time limiting step in developing a vaccine is in human testing. Can we use 21st century technology to minimize or accelerate these steps?
Well, yes. Well, first of all, I'm not a vaccinologist or I haven't worked in a vaccine development process, but I think what those folks would say is that this process so far has been the fastest by far of any vaccine developed in the history of humanity, and that they have used platform technology and particularly the things that are out leading the front are mRNA technologies, which while they haven't been used to make vaccines successfully in the past, there's been a substantial amount of investment in that technology. And so within about 65 days from knowing the sequence, the first trial was able to be initiated, which is really an incredible timeline. So I think there have been lots of technological advancements that have helped accelerate it to the process that we now are seeing. But my hope is that we can continue to find new efficiencies, and in preparing for the future, we might be able to shorten it even further.
One of your audience members had asked if you could speak a little bit more on international collaborations that you have witnessed that have been effective.
Sure. Yeah. I mean, there are countless examples in the world of international collaborations. There have been extraordinary collaborations, going back to the smallpox eradication program or the Rinderpest program. Those were incredibly successful programs for humanity. But those are, there are, on any given day, there are international collaborations around malaria, tuberculosis, on yellow fever. There are all number of international organizations which are aimed at diminishing global health burdens, and they get their resourcing from foundations and from governments, and they usually have people from around the world working in those organizations. There are also multilateral initiatives that country to country might take on, even in the absence of an international organization. But my view is that the U.S. should be a strong part of the international global health community, and that has been really important for the U.S. has been a major contributor to global health collaborations in the world for generations. It's been a recognized leader in those collaborations. And in return, I think we learn an enormous amount. We have collaborations on the ground with researchers from around the world. And enormous goodwill created between countries as the U.S. is trying to partner with and find ways to help other countries. So I think, yeah, there's kind of a limitless number of examples of global health collaborations around, endemic diseases and during emergencies.
Paula, is there another question?
Oh, there's plenty more.
Why don't we take one more and then move on to Professor Charo?
All right. So I have several questions that have come in about kind of returning to our routine lives, sending our kids back to school or returning to work, and in situations where social distancing is quite challenging. And folks are asking about, should they wear an N95 mask? What PPE should they be using to protect themselves and their coworkers and their families?
Well, first thing I'd say is that the CDC and state health agencies have a huge amount of information that's very specific about the kinds of cloth masks people should use, the advice on social distancing, lots of advice on schools. So I would start by just referring people to those resources. There's lots of valuable information there, not to mention many other places. And I really won't try and summarize it here. But in general, I do think it's going to be some time before we get back to what we would consider pre-COVID life. but I think it's possible for things to keep getting better incrementally over the course of the fall. And it's certainly when vaccine assuming we do get a safe and effective vaccine, and I'm hopeful we will, we'll begin to see people vaccinated over time. And I think it will be over the course of the winter when I hope they begin to be occurring in larger scale. And so we'll begin to gradually get a little bit more normalcy over time. But in the meantime, I think we should strive to get incidence down in our communities so that we can get closer to like a experience of many European countries, which have very low incidence. They haven't eradicated the disease, but they're doing pretty well. Similarly, Iceland, Singapore, many countries in Asia doing very well. We could get there, not overnight, but if people really worked hard with masking and individual physical distancing decisions and smart avoidance of large gatherings where we have super spreading events, I think we could make a lot of progress down the road.
Great, thank you, Tom. We'll get back to some of those questions at the end of the day. Thank you so much for the presentation. We'll now turn to hear from Olta Charo. Professor Charo is the Warren P. Knowles Professor of Law and Bioethics at the University of Wisconsin. And as most people in Washington know, if there's a question of governance of an emerging science or technology field, the person you call first is Olta. Professor Charo.
Well, I'm a little scared now that you've advertised my availability. Thanks very much, Anne Marie, and thank you to Los Alamos and to Texas A&M for the opportunity to speak. I want to talk about something that is, I hope, going to be a change in the way in which we think about bioethics in the coming years. Next slide, please. For those that are unfamiliar with it, medical ethics has tended to focus on the doctor-patient relationship and on the patient, him or herself. As a result, a lot of the ethical issues have focused on the degree to which we have autonomy over decisions about our own health, even when those decisions might not be in our own best interest from a medical point of view. It tends to focus on the duty of care, particularly from physicians, and the duty to provide information to allow people to make decisions for themselves. When it comes to questions of justice, again, it tends to be very individualistically focused about whether or not you're discriminating against somebody because you don't like their politics or because you're uncomfortable with their ethnicity. And the success of health interactions tends to be measured in terms of good medical outcomes, or if not that, at least a good outcome in terms of patient satisfaction in the form of having exercised autonomy. So it's very individualistic. Now, this is all very general, and certainly in medical ethics over the years, we have seen more and more incorporation of other kinds of considerations. But fundamentally, medical ethics is very individually focused. When it comes to public health ethics, however, we have a field that's much less well developed. After all, medical ethics began primarily with a kind of triad of doctors and philosophers and theologians, which did not really lend itself to a public health ethics, where the distinctions and the questions are different. Instead of a focus on the patient, the focus is on the population as a whole. That might mean that an individually tailored intervention, lowering your salt intake, might make sense for one person versus another, but at a population level, you might decide you just need to give that advice out to everybody, because most people won't be hurt and some people will be helped. It's very pragmatic and it's very utilitarian. There's also a different balancing on the issue of autonomy. In medical ethics, really the concern is about being able to express autonomy over my own condition. But in public health ethics, we worry about whether that autonomy is going to work to the disadvantage of others. And that's exactly what we see in the area of infectious disease. It's exactly the kind of thinking that underlies vaccination in order to develop things like herd immunity. The focus on the duty, instead of being the duty of the caregiver, then becomes a focus on the reciprocal duty of the community or its representatives, the government. If there's a sacrifice that's being asked of the public, such as lockdowns, or any kind of quarantines, then there is a concomitant duty to relieve the sacrifice as much as possible, to replace the opportunities for lost income, to assure adequate food, etc. Because to ask for sacrifice on the behalf of everybody generates a reciprocal duty for everybody to try and minimize the cost to individual people. On justice questions, we get into the area of whether or not the benefits and the constraints are being fairly distributed. This particular pandemic has brought to the fore in a way that has never happened before exactly how acute our health inequities are in the United States, starting with which people have living conditions and employment conditions that put them at higher risk, coupled with living conditions that make it easier for them to transmit those risks to others, the comorbidities that develop because of their living conditions, and the absence of equal access to health care, whether through insurance or just the way the medical system has treated them as individuals. The result being that we have dramatic differences in the degree of morbidity and mortality as among disadvantaged minority groups and the rest of the population. Differences as much as 3 and even 5-fold when we talk about African Americans and Alaskan Natives and Pacific Islanders. Last, in terms of these kind of big picture differences, is that instead of measuring the outcomes solely in terms of medical benefit, in public health ethics, we also look more broadly at societal outcome. And here, the cost of these kinds of public health measures in the form of lost social functioning or poor medical outcomes because of the absence of preventive care. Just today, studies out at the number of stillbirths that have increased, not because of COVID infection, but because high-risk pregnancies are not seeing the physician as often as they would, are the kinds of things that get rolled into a more holistic view about outcomes in public health ethics. This means you need much more attention to things like economics, political philosophy about the role of government, anthropology, sociology, and psychology to understand the way human beings react in order to develop a rich and textured view of how a particular intervention is going to affect not only individuals, but the entire community. Next slide. So for public health, we tend to focus on several different kinds of interventions. Primarily, we start with self-protection. That's the classic hand washing, where if people don't want to do it, they tend to say, I'll take the risk for myself. If anything, this is the nanny state problem, where the public health authorities tell you not to drink too much soda, or they want to have soda only sold in smaller cups, because we've learned that people don't bother going back for a second cup, and so it has the effect of lowering your sugar intake. And it tends to run up against an American libertarian, autonomy-based kind of instinct that makes even these seemingly benign measures very controversial. Next slide. But it gets much more complicated when we ask people to take actions that are not entirely for their own personal benefit, but for the benefit of others. And so we have here one of the many, many wonderful images I found of the protests against the many, many different government efforts to stem the pace and severity of this pandemic. The Bill of Rights doesn't have a virus clause. These are people that are going to be protesting against a variety of specific measures. And I'd like to talk about some of them in particular. Next slide. One of the first key controversies is masking. And so most of the people in this audience understand that while a mask offers some protection for you yourself, the primary reason that we were asked to all wear masks was to make sure that we didn't pass the infection on to somebody else. In a disease that has a long latency period that is associated with transmissibility, or a disease like this one, where many people are infected but are completely asymptomatic and unaware of their infection status, masking to protect passing it on to somebody else becomes a crucial public health measure. So it really becomes a question of how much we ask you to do for others. You'll notice that at the bottom, I don't know if you can quite see the picture, the sign that the woman is holding is, "Your fear doesn't take away my freedom." A direct challenge to the idea that we should be able to require sacrifices of individuals on behalf of others. Now, this is something that goes way back in our history. You know, over 100 years ago, there was a seminal case that Judge Tatel mentioned in his introduction Jacobson versus Massachusetts, about inoculating people in order to protect the community from smallpox. Masking is certainly a less intrusive measure than actually vaccinating somebody. And yet even this seemingly simple measure has run into tremendous level of resistance because we have not yet completely transformed the notion of civil ethics and public health ethics into our kind of daily vocabulary. And instead, when people think about bioethics, they are still thinking about the doctor-patient relationship and thinking only pretty much about themselves. Next slide. Another key controversy has been gatherings. This one gets much more complicated when it comes to civil liberties because you really do have specific provisions in the Bill of Rights that go to the question of the freedom to assemble for any purpose, whether it's to have a party or to have a protest. The freedom to worship, which for many people involves going to a particular place that's associated with worship. Or freedom of speech, again, something that comes up particularly in the protests like we see here with the Black Lives Matter. So in all of those areas, we now have the beginning of a true constitutional challenge to public health efforts to shut down large gatherings or to limit the number of people at any one time. Again, Judge Tatel mentioned a recent case in Pennsylvania, Butler versus Wolf, which just came down in the last day or two, challenging Pennsylvania's variety of measures to have people shelter at home, to close down some but not all businesses by trying to distinguish between those that are essential for social functioning, like groceries, and those that may not, like hairdressers, as well as a variety of its other measures, including restrictions on the number of people who could gather indoors at one time in certain settings. And the irony of the case having to come up in Pennsylvania is that in 1918, when it was the 1918 Spanish flu that was now circulating in the United States, it was Philadelphia that chose not to cancel a massive parade. It was a patriotic parade about the end of World War I, but it was 200,000 people marching through Philadelphia. They chose not to cancel it, and one of the results was that Philadelphia had just about the worst experience with flu infection spread, morbidity, and mortality as among all major cities in the United States. And Howard Markle, who was mentioned just by Tom Inglesby, was in fact one of the people who studied that phenomenon. So here we have Pennsylvania once again at the forefront of deciding whether or not to take the rather extreme, admittedly extreme actions necessary to try and slow the spread of this pandemic. And in this case, the court found that the justifications, although well-intentioned, and it went on to emphasize well-intentioned, were simply not sufficient. They either were not tailored narrowly enough, they were overly restrictive when they should be less restrictive, they didn't have clear endpoints as to when those restrictions would shut down. They made seemingly unexplained distinctions in how they decided which businesses were going to be essential or not. And the result is that at the moment, the Pennsylvania efforts are stymied. This case will certainly go on, and there have been other cases in other jurisdictions looking particularly at things like gathering in churches for worship, where again, First Amendment privileges about religion are being countered by the claim that public health needs these kinds of restrictions. But we are really very fundamentally seeing a challenge to the role of government and the degree to which we have to sacrifice in order to protect everybody's health over the long run. Next slide, please. Part of the challenge in public health is going to continue to be figuring out how it is that we're going to define this common good that we're setting up as in tension with some of these freedoms, whether constitutional or simply cultural. Some efforts at this, such as in Minnesota and at the University of California, San Francisco, tend to be things that are focusing on process, transparency, accountability, trying to make things available to people so that they can debate them. If you go to the statement that Johns Hopkins made in August of 2020, and this is as paraphrased in a draft from the National Academies on vaccine allocation. That draft went out about 2 weeks ago. It's now in revision and will be published in final form in a couple of weeks and provide advice on how to allocate the limited supplies of vaccine that Tom was talking about. Here, the effort was to really define common good in a broad way, that it's preventing morbidity, preventing mortality, protecting the integrity of the health systems that might otherwise get overwhelmed by demands for ICU beds or ventilators, but also promoting economic and social well-being, which means keeping essential services going and, frankly, figuring out how to educate our children, many of whom are terribly disadvantaged by the absence of the public schools, since they do not live in homes where they have parents who can substitute as teachers, or where they have a quiet place or an internet connection that allows them to take advantage of online opportunities. So the very question of how broadly you define the common good may in turn relate to what level of sacrifice we are going to be able to justify and how those sacrifices will be distributed. Next slide. On the closings, one of the most difficult things to figure out is how we can calculate the larger cost of slowing the spread of the virus. There have been people who suggested at the earlier stages of the pandemic that we simply should allow for the society to stay open, and those people who are vulnerable or older vulnerable because of age or comorbidities should simply take extra steps to protect themselves, even if it means that they lose their jobs or in fact wind up dying because the cost for collective restraint was too high. And we are certainly seeing a tremendous cost, not only in terms of the education of a whole generation of students, but also in terms of the permanent loss of some very big, well-known brands. So we see a large number of bankruptcies. We see people who are running out of unemployment insurance and special unemployment provisions, who are now at the point of not being able to afford food or rent. We see landlords who are going broke because their tenants can't pay the rent. And so a real challenge in any of these things is to figure out exactly how broadly we're going to try to calculate those social costs in this very delicate balancing act. But at the end of the day, for public health, the goal is to try and figure out what that balance might be. Next slide. Key to all of this is how both the benefits of slowing the pandemic and the costs of the shutdowns have disparate effects on disparate populations. So bioethics post-pandemic is, I think, not only going to be struggling to broaden its view to encompass a wider notion of the common good and the kind of secondary and tertiary effects of various kinds of health policies, but is absolutely going to spend far more time than it ever has looking at health disparities and their effects on individuals and whole populations. Again, bioethics has had people thinking about issues revolving around race and socioeconomic status for years, but not at a level of intensity that we are now seeing as conference after conference and webinar after webinar has now been announced in order to emphasize how it is that we should be thinking about these kinds of disparities. The confluence of the pandemic and the Black Lives Matter movement has certainly made it impossible to ignore this. And it's made it impossible to ignore the question of whether a seemingly neutral policy or the neutral development of a new technology can ever be analyzed against the backdrop of fundamentally unequal background conditions. And this has been a challenge always when looking at how we should pursue a new technology, whether it's genome editing or a new kind of diagnostic test, having to force ourselves to ask much more actively, how can this be developed from the outset and rolled out in a way that minimizes the possibility that it will simply continue to reify these disparities that already exist? Next slide. Next slide. Thank you. We are already beginning to see some academic literature developing around that. SSRN is the Social Science Research Network. It's the kind of preprint location for people in the social sciences. And every day there are additional articles that are focusing on exactly this and trying to analyze this at a national level and at a transnational level. Last slide. So to summarize, bioethics after the pandemic, I think, is going to move away from what had been an early kind of focus on autonomy and beneficence, and is going to move toward a focus on justice questions. Much more focus on public health ethics as opposed to individual doctor-patient, much more attention to structural issues in the healthcare delivery system and in the underlying living conditions that generate disparities in the kind of upfront health status that individuals have. It's going to force us to have a deeper view about what civil rights mean and how and when they can be limited to some extent for common good. And to do that, a lot more attention to areas that have been somewhat on the edges of bioethics for a long time. Relational ethics, focusing on the ways in which people are technically autonomous rational beings, but in fact are deeply influenced and constrained by the people around them and the relationships they have with them, whether it's their parents, their children, or their colleagues. Feminist ethics, looking specifically at the way in which the role of women is understood by women or imposed on women, affects the degree to which they will think through problems differently or have solutions foisted upon them. And critical race ethics, which looks at the structural forces that change how it is that various kinds of marginalized communities will view everything from their trust in the medical system and enrollment in clinical trials to the way in which they do or do not comply with medical advice or offer to sacrifice themselves for the sake of others. I think this is going to be a moment where we are seeing a transformation, and a welcome one in the lens through which we view ethics and medicine in the United States. Thanks very much.
Thank you very much, Olta. I know there's a number of questions, but I wanted to just pose one to you that came up earlier with respect to vaccine allocation, because I know you've been involved in a number of efforts to think about that, and perhaps you could fill us in a bit on the current thinking on that.
Sure. By way of just telling you where I'm getting my information from, I'm on the National Academies Committee that's writing a draft framework for vaccine allocation that will be advisory to the CDC. And I'm also on the corresponding committee here in the state of Wisconsin. The anticipation is that whenever the vaccines begin to come online, and there is great hope that they will, that the initial supply of vaccines is going to be extremely limited. And so there's necessarily going to have to be a question of who goes first and who goes second. If you look across the history of various kind of crisis standards of care and rationing systems, you see some commonalities, but each one has some peculiarities due to the particularities of the disease. In this case, what we're seeing is a kind of convergence around the idea that you're going to probably have to have healthcare workers, not only physicians and nurses who have direct patient contact, but the other people, the transport workers and the orderlies and others who have direct patient contact, probably come very, very early on and getting vaccinated because they were at both higher risk of catching the infection because of their contact with infected patients, and also are crucial to maintaining the healthcare system's ability to continue to serve the rest of us. After that, you tend to get a variety of efforts to look at the following kinds of factors. Who's at most risk of being exposed? So you're looking really at working and living conditions. Who's at highest risk of getting seriously ill or even dying from that if they get infected? So you're looking at underlying comorbidities and age, which itself is an indicator. You're looking at which people are working in settings that absolutely must be maintained at full strength in order to manage societal functions that are essential. And that can mean a lot of very, things that you don't think of as being high profile activities, the delivery people, the transportation people, the cleaning staff, and which people are going to be in some setting where their ability to take care of themselves has been restricted. That could be congregate settings like prisons or nursing homes where you lose some amount of autonomy. And then the phases for vaccine rollout tend to be aligned along these criteria. One significant debate has been whether to identify minorities, particularly because of the dramatically different effect that this pandemic has had on them. It turns out that while there is a disagreement about whether to name by minority group or not, that the kinds of criteria that I mentioned earlier tend to have an effect to mitigate inequities, because the people who tend to be in the most highly exposed areas are the people with person-to-person contact. That tends to be service workers and others who can't just remotely work from home. And that is disproportionately minorities. Caregivers and in-person caregivers at nursing homes or for the elderly in their own homes, again, disproportionately from those populations. Comorbidities, more frequent in the marginalized population. So again, moving up on these criteria toward the head of the line. So whether you do it through looking at these kind of race and ethnicity neutral criteria that have the effect also of identifying people in minority communities, or you name minority communities, either way, most of the proposals are at least tackling the dilemma of a pandemic that doesn't hit everybody equally and trying to figure out a vaccine rollout that will help deal with that problem. Last but not least, I know I'm going on, sorry, Anne-Marie. Whatever you want to do in terms of phasing things in, you're going to confront two very big obstacles. One is that logistically, some of the vaccines are going to be very difficult to get out into the communities because of some of the requirements for their safe transport and storage, particularly those that need to be stored at minus 80 degrees centigrade, so-called cold chain problems. The second is vaccine hesitancy, the percentage of people who simply won't get vaccinated. This is above and beyond the kind of vaccine hesitancy that we see already growing to a distressing degree in the United States, particularly for pediatric vaccines. But now added onto it are surveys like the one from the Kaiser Family Foundation documenting a growing distrust because of the perception that politics may influence the way the vaccine trials are conducted or their results are interpreted or the FDA acts to approve or not. So surveys are showing between 30 and 50% of people saying that they would be hesitant to take such a vaccine. If that happens, then the phases that we've talked about in all these different settings are probably going to be really inadequate to address the kind of problem of getting the vaccine to the right people at the right time. And we'll see a tremendous distortion in the way in which the rollout occurs. Sorry, I know that was a long answer, Anne-Marie, but it's a complicated topic.
Thank you, that was very important. Paula, do you have another question?
I do. How will the COVID pandemic impact the way societies make decisions about future pandemics, especially with regard to fairness, justice? Will it have the same impacts as it is having with the field of medical ethics and bioethics?
So the question, I'm sorry, the question is how will it affect pandemic planning as opposed to post-pandemic ethics? I'm sorry.
So I'll try to restate it. How will this pandemic impact the way future societies make decisions about future pandemics with respect to fairness and justice?
I guess that's what I was thinking of ethics, is fairness and justice. That's why I was somewhat confused. Yeah. I do think that in terms of pandemic planning, which includes fairness, justice, but also governance and logistics and everything else, it's, I think, going to teach us to, first of all, not abandon the very excellent efforts that were made in the past, like the Pandemic Preparation Office that had existed within the White House until the last couple of years. Second, it's going to sensitize us, I think, to the global issues because of the transformation of the American position vis-a-vis participation in the global community's COVAX program and other sharing programs. You know, we are facing right now a concern about growing vaccine nationalism. And I think this is something that we haven't really faced that much before because we've rarely had a pandemic that was this globally distributed. Usually they're more focused, and that will probably start a new conversation about our obligations not as individuals to each other, but as a country to other countries in a global effort to control pandemic spread.
Terrific. Paula, are there other questions there? Well, you're looking, Alta, perhaps you could say something about your description of the bioethics and public health ethics, medical ethics and public health ethics really was sort of framed within the context of the US in many ways. And you've been so engaged in global discussions about ethics and governance. I wonder if you could say something about that, whether there are things we can learn from the other approaches that other countries might take to thinking about ethics in this context.
Well, it's funny, I was just reading this morning a piece written by a neurologist from New Zealand, in the University of Otago, who himself suffers from a variety of medical conditions that puts him in the frail category, as he put it. And he was contrasting the New Zealand and American experiences. I think that there's both cultural and constitutional reasons why the US is unusual as a member of the world community. In many countries, you simply do not have the same degree of presumption of individual liberty. And so government actions can start from the premise that the government can say what is allowed or not allowed. And it's really only controlled politically. Whereas in the US, we start from the notion that the government is one of limited powers, that it must have a specific identified power in order to act, and outside those areas, the presumption is individual liberty. And that really is part of what accounts for the very different experiences we've had, specifically with things like the lockdowns, where in places like France, everybody was locked down unless they had specific permission to go out. And it was even enforced on the streets by police officers who could ask to see your paperwork that said you were one of the people allowed to go out. Contrast that with the US, where the enforcement is much spottier and much more focused on voluntary behaviors. I'm not saying that the US is an inferior culture, far from it. I grew up here, I kind of like it. But it does mean that we need different strategies than you would have in countries where the role of the government is a little bit more centralized and more pervasive than it is here.
Terrific, thank you. Paula.
Do you mind if I move to some general questions for the panel?
To bring Tom in also to the discussion, sure.
Where do you see the bottlenecks in the US now with respect to testing, tracing, or public social behavior? And how do you recommend that or suggest that we deal with it? And your views on the country reopening?
A lot. You want me to start? I'll just say a couple things about it. I think contact tracing is still a very difficult enterprise in the country. Some states are doing better than others, but overall, we haven't gotten to the numbers that most experts believe are going to be necessary. It's also not possible to do contact tracing very effectively when huge numbers of new cases are appearing in a particular state. So we have to get the numbers down through other kinds of policies and then use contact tracing as kind of more of a precision tool. Diagnostic testing has gotten better, but the turnaround times in some places are still quite slow. And we are going to see in the fall the coming online of rapid antigen tests. Many companies are making them. They're getting better at them, and there may be different ways of doing testing, which are more distributed and may not be as precise as PCR, but would allow people to do testing in institutional settings, and maybe in a perfect world may help us identify contagious people more effectively and be a little bit more precise about what we're doing.
Paula, we're waiting for another question. Perhaps I could pose one to both Tom and Alta. Tom, you mentioned a countermeasure program, and it involves academia and government and industry. And Alta, you talked about vaccine allocation, vaccine development and allocation. and obviously involvement of the same players. And I wonder if you think that in the midst of a pandemic, we have the right framework in which industry and government and researchers can work together, or whether we're seeing that there are areas where there's too much rigidity, or there's not enough sharing of data, or IP gets in the way, and we need to think differently about the way those entities interact with each other.
Alta, do you want to start?
No, you start first, Tom. I'll go after you.
Well, it's difficult. That's a very big question. I think the U.S. government has spent a lot of money trying to create relationships with industry in various development programs and contracts. In BARDA, particularly, that's the name of the agency that does the most work with industry. NIH has incredible relationships with the universities around the country. Whether or not we're harvesting or getting the fruits of all the university science into industry in the most efficient way, I don't know. I'm sure we could be doing better. But I do think that there is the U.S. has an incredible scientific community, and in both in academia and in NGOs and in industry. So I think you're right that we should try to keep moving in the right direction and improve the interactions between them, and see what we can learn from this pandemic.
Yeah, I would add a couple of things specifically about the relationship between government and industry when it comes to developing new therapeutics. One of the things that's interesting about Operation Warp Speed was the way it tried to de-risk the financial investment that companies make in the development of a therapeutic. Instead of having them invest tremendous amounts of money and then incrementally continue investing as they see incrementally suggestively positive results, which means that you get a very slow ramping up. And then at the end of the day, if something gets approved, the companies want then to have a long period in which they can make back their money and a profit by having exclusive rights to sell at a higher price. Instead here, what the government has done is that it basically financed the ramping up to scale for scale manufacturing before we knew if a particular vaccine was going to be successful. That means that it's the government that took on the risk the vaccine would fail and its investment in scale up would be lost. That de-risking allows for companies to be far more adventurous in how quickly they try to move. And hopefully in the future, it will provide a model for something that would also allow for more upfront investment in therapeutics that then come on the market without the kind of elevated prices that are part of the business plan that we see now for most drugs and biologics. The second is a fair amount of sharing of data, which meant that you had to have intellectual property agreements that would allow for kind of a collectivization of the intellectual property and the data so that everybody moves forward more quickly. And third, clinical trial protocols, where you got more and more emphasis on adaptive trial design that will, throughout the trial, continue to perhaps change the various arms of investigation or the number of people allocated to each as the data is rolling in real time so that one can try and hone in on a result more quickly. And there's already conversation taking place, even just, again, you stick today, I spent a lot of time reading news, a piece by leadership from some drug companies talking about wanting to somehow gather these lessons and see what can be maintained at the far end of this pandemic, so that we don't return to business as usual, no pun intended.
Great, thank you very much, Alta. Paula.
I think we're starting to dwindle on our audience questions, but I think this is a good segue. In terms of what we expect to see in terms of the illness as we move into the fall and winter, what do we anticipate, or what do we believe is what we're going to see with respect to COVID-19 and the combined effects with influenza?
I think that's you, Tom.
Yeah. So yeah, sure. So the question is combined effects with influenza. What do we expect? Is that the main question, Paul?
That is the main question.
Yeah, I think hopefully we will have a similar experience as the Southern Hemisphere is having, which is unprecedentedly low influenza activity, We've seen that in Australia and South Africa and other countries that are reporting, and it's presumed to be because of the very dramatically diminished social interaction that we all are living through. And so they've had a very, very, very mild flu season, which is the opposite of ours in the Southern Hemisphere. So we will hope to have that kind of experience, but I don't think we should plan on having that experience because we don't know how leaders will be leading us in the next few months and over the wintertime. We don't know how the public will be reacting. So CDC has announced that they have an unprecedented amount of flu vaccine available this year, and public health authorities around the country and physicians' offices are all encouraging flu vaccination. We should be doing whatever we can to try and decrease the incidence of flu, because if we do have a bad flu season and we do have continued surges of COVID-19 in various places, that could be an extraordinary burden on ICUs and hospitals and healthcare workers. And also it could lead to lots of diagnostic test challenges, delayed in testing times as everyone goes and gets their every fever and every flu symptom checked out with both influenza testing and COVID testing. So hopefully we'll do better than usual, but I think we should plan for a serious flu season in any event and do what we can to prevent that.
Thank you very much, Tom, and thank you to both you and Alta for your wonderful presentations and for your thoughtful responses to these questions. Paula, over to you now.
Thank you all, Alta and Tom and Anne-Marie. Thank you for all of your insightful comments. I'm going to turn the webinar over to Diane Hurtado of Texas A&M University for the closing remarks.
Hello, everyone. We hope you enjoyed today's presentations. Please join me in thanking all the moderators and speakers for their outstanding presentations and discussions. I would also like to thank the organizing committee and especially Rajan Gupta and Paula Nepper for their leadership. The tireless efforts of this committee allowed us to pivot from our original face-to-face symposium to the topically relevant webinar. And since we did have to move to a virtual platform, we had the opportunity to learn a completely new skill set, and we greatly appreciate the team at Texas A&M and Los Alamos who managed this event, namely Adriana Abreu, Michaela Catalina, and Tanya Wolf. Without their hard work and problem solving, we could not have enjoyed this event today. We hope that all of you will join us for the future Harnessing Transformational Technologies events. An email will be sent to all that registered for this webinar with a link to the event recording, an opportunity to provide feedback and info on our upcoming webinars. On behalf of Los Alamos National Laboratories, the National Academies, and the Texas A&M University System, thank you very much for joining us. And that's all I have. Bye.